Welcome, everyone. Thank you for joining us today. I'm Marina Hatsopoulos, and we have a great program ahead on COVID-19 genomics and its outlook. The Hellenic Innovation Network, with the support of the Greek Consulate in Boston and MIT Enterprise Forum Greece, hosts events to educate those in the United States about innovation and entrepreneurship taking place in Greece, and to offer Greek entrepreneurs the opportunity to pitch a network within the very rich Boston tech ecosystem. We also have a CEO group which meets monthly in order to exchange best practices and to help support each other. And finally, we have a platform at hellenic.org where you can register and network virtually with other members of our community. Due to COVID, we've been unable to host our in-person events, although we hope to do so at some point soon. So we've been doing a Zoom series relating to Greek technology entrepreneurship. And this is the last session of our series for this season. We'll be starting again in the fall in September, and we hope you will join us then. So for today's event, please email any questions you have, and any that we are not able to get to due to time constraints, we will post on hellenic.org. Without further ado, I would like to introduce the Consul General of Greece in Boston, Stratos Efthimiou. Stratos.
Thank you, Marina. I'm personally thankful to you and to Vasilis and all the members of the Hellenic Innovation Network who have worked tirelessly for this unique series of webinars, which deepened our network and strengthened its echo. I would like to extend a warm welcome to Manolis, Kellis, and Katia Karalis, two very human and truly special researchers and academics. I would like to thank them both for their continued support, and I would like to wish congratulations to Katia for the new page in her professional career at Regeneron. Professor Kellis is an exceptional academic, a shining young mind who has always been giving back to his community. I had invited him to give a lecture in early March at the Consulate General, a lecture which we postponed as early as February. I believe we were the first in the Greek community to postpone an event, and I would like to thank Manolis for this very wise piece of advice. He tells a lot about his social responsibility as a scientist. Had we proceeded to this lecture, it would have become a super spreader event disseminating the virus in the Greek American and bioscientific community here in Boston. So the following day after we canceled our event, Manolis posted publicly a thoughtful text which was shared by thousands of people informing them about COVID and what we should and we shouldn't do. I think these two incidents tell a lot about Manolis' character and his belief that science is not only for scientists and labs, but it is rather a means to serve the advancement of humanity. With this, I would like to invite Vasili Papakonstantinou, whom I thank for his precious help. Thank you.
Thank you, Strato. Thank you, Marina, Katia, and Manoli. Hello, everybody. Thank you for joining our webcast today. MIT Enterprise Forum Greece is a non-profit organization that helped form Hellenic Innovation Network. And it has a mission to support technology entrepreneurs here in Greece and help them turn their startups into global companies. Our flagship program is a startup competition, and we are currently in the final stages of the competition's 6th edition with great teams, some of which are coming straight out of Greek research labs, which is very encouraging and very interesting because it connects very much with the contents of our discussion today. You can follow us on social media. Keep an eye for the announcement of the finals event that will take place in mid-July. Our moderator today is Dr. Katia Karalis. Katia is a trained genetics and experimental endocrinology with broad experience and contribution in basic and translational research, as well as strategic, scientific, and managerial leadership positions in academic and industry settings. She focuses her research in understanding how disturbed homeostasis of the organism, such as when exposed to external or internal challenges, drives the disease mechanisms and how we can overcome the insults with new therapeutics. She has a solid experience in inflammatory disease, immune response, stress response, metabolism, organoids, and organ chip technology. She recently joined Regeneron Genetics Center to lead the biology group for the understanding of new mechanisms for genetic, inflammatory, metabolic, and neurodegenerative diseases and develop new drugs for human patients. So, Katia, welcome. The floor is yours.
Thank you very much, Vasily. Thank you, Strato and Marina, and Manolis for agreeing to participate in this discussion. And Manolis is a computer science professor at MIT, and his whole career is focused in understanding genome and all the variations in genome that are related to diseases. And he will bring this knowledge in the context of COVID. So, Manoli. Hello.
Hello, everyone. So thank you so much for having me. Strato, Marina, Vasily, you guys are an amazing team. And I'm so honored and humbled to be part of this series and to follow just this incredible series of conversations that you've had so far, which have been really illuminating, not just in the quality of the discussion, in the quality of the science, but also in the bonds that they form within the Greek community. So it's a real pleasure to be here, and thank you, Katia, for hosting me. It's a real pleasure to chat with you, both in our impromptu conversations, in our scientific conversations, and in this forum.
Thank you, Manolis. So we'll jump straight to the questions, and so we'll have some more time, save time for the questions from the audience. So as I mentioned in the introduction, where we focus today, it is you as a genomicist and COVID-19. So tell us about your perspective on and how you started working in the COVID virus as soon as we all realized it is something more than the common cold.
So basically my work has focused on genomics. So basically understanding genomes mostly of human, in order to understand how genetic variation leads to disease. And my group has basically studied in an unusually large number of diseases between Alzheimer's and schizophrenia and cardiac disorders and immune disorders and cancer and obesity and diabetes. So we've basically tackled many different disorders. And the reason for that is that genomics, in my view, is the glue. It's sort of the thing that sort of unites human disease. And the way that I like to describe it is that the advent of the central dogma, the recognition of sort of DNA mix RNA mix protein and all of its exceptions, unified biology. Instead of having zoology department, botanology department, and virology department, you suddenly had biology departments. And I think in the same way, the genome and genetic variation is effectively a unifying force behind the manifestation and the causal understanding of disease. So the methods that we have developed in my group really apply to a very broad set of disorders. And that's why we have established collaborators with each of these different fields to really make a difference in understanding how are genetic variants functioning. And we've brought a series of tools for understanding how genetic variants function. The very first set of tools is also what I'm going to be talking about today, which is comparative genomics. Basically, we've brought in the lens of evolution as a way to understand genome function through the mutations that have happened across mammalian genomes, across 60 million years of a mammalian evolution. We can basically see countless evolutionary experiments of perturbing this nucleotide and that nucleotide and that nucleotide. And through these experiments, we can understand what are the basic functional building blocks of the human genome. Where are the genes? Where are not genes? Where are circuitry elements that govern how genes get turned on and off? And these tools, this arsenal of tools, has basically painted a picture of about 5% of the human genome that is clearly under nucleotide level evolutionary constraint that you can use to now start pinpointing where are the functional elements. So basically what my group does in each of these collaborations is study systematically how are mutations in the human genome perturbing these functional elements, and how are these elements then leading to function. And of course, that's only one lens. We also have the lens of epigenomics that tells us which of these elements function in different tissues and cell types across the human body. And we've used that to sort of recognize that, for example, Alzheimer's genetic variants don't localize in neurons, but instead in microglia, in the immune cells of the brain, shifting the focus from a neuronal disease to a really immune disease and inflammatory disease for Alzheimer's, which has major implications in the fight against Alzheimer's. Through the same methods, we basically showed that obesity, instead of sort of controlling the circuitry of what do we choose to eat and whether we choose to exercise, the strongest genetic association with obesity is in fact instead controlling our fat cells to decide how many calories to burn or to store after a meal. And that basically determines the vast majority of the risk, of the genetic risk for obesity, which basically sits in this FTO locus. So that's what we've been doing, and we've been unwittingly dragged into virology over the last few months in the context of COVID-19.
So since you mentioned your other work with human diseases, we'll get back to the virus itself, but would you like to discuss what we learned? It is not a long experience. A lot of people were affected and a lot of studies at all over the world at the same time. That was amazing scientific effort. But what did we learn from the differences of the human responses to the same virus?
So the responses have been dramatic across individuals of different countries and across individuals in the same country. And there's a lot of factors that contribute to that. The first and foremost is social isolation. Basically, how fast did a country actually follow the scientific guidelines of isolating, of not having large meetings, not having large gatherings, which have been extremely detrimental. So basically, there's been, for example, the dramatic response difference between the UK and Greece. Both of them isolated eventually, but Greece did it after a very small number of cases, and the UK did it after thousands of cases. And the reason why this response is dramatic is because there's a lag between the time that you discover that things are really bad and the time when you made the decisions that made things become really bad. Basically, because of the two lag, interval between the time that a person first contacts the virus and the time that they are in the hospital fighting for their life, there's a 2-week lag. That basically means that as the number of hospitalizations is going up, it's already too late, because if we react then with a virus that was doubling every 2 to 3 days back in March, we basically have, 3 days of response means twice as many deaths. 3 more days of lag in response means 4 times as many deaths, and so on and so forth, 8 times after 3 more days. So if you wait for 2 weeks, you basically see differences in the, hundreds of thousands of infections versus only tens of thousands just by this small lag. So to come back to what are the factors, basically number one is how fast did the country respond? Number two, was the country in direct proximity of China? And this has affected the delay. This has resulted in the delay that we see now in Latin America and in Africa, where the virus is unfortunately exploding right now with many weeks of delay compared to the countries that have more economic ties and more travelers and more large events from people traveling to China. And I think that's where the differences between Greece and many of the other countries come in. Basically, number one, we took very early steps, and I want to praise the Greek government for really letting the scientists do the talking, for really following the scientists' advice and implementing this advice in guidelines for the entire population. And of course, the population of Greeks, because it takes great followers to make great leaders actually shine. And without the entire population of Greece basically being very strict about this, I think Greece wouldn't have much to celebrate right now. So that's one aspect, the response. The other aspect is, of course, that Greece was a little delayed compared to direct travelers from China. And then there's other aspects that have been sort of floated around. For example, there's a very strong correlation between vaccination policies at the country level versus number of deaths from coronavirus. And it is possible that cross-reactivity or just simply boosting of the immune system from early childhood vaccines may have actually played a role in variation. And then the last part is inter-individual variation. Basically, many individuals contact the virus, not every one of them gets sick. So of course, age is the number one factor by far. The elder a person is, the risk increases tenfold. Obesity, which is unfortunately a big issue in the United States. There's aspects of male versus female differences, asthma, history of respiratory problems, history of cardiac problems. And lastly, genetic variation in the actual genome. And the first genome-wide association study basically found two very, very large hits, and both of them point to immune function. One is, of course, blood group, and the other one is other related immune genes. And variation in the immune system is something that we should embrace as part of not just the human condition, but part of life. Basically, any species has a strong selective pressure to maintain a diverse immune repertoire so that a virus doesn't wipe out the entire population. The closer individuals are genetically within a species, and I think the cheetah has been a prominent example in the popular literature, the more likely that species is to fall prey to any kind of virus that has adapted to any one of the members of that family. So right now, the SARS-CoV-2 virus has indeed been extremely well adapted to human-to-human transmission, but the way that it wreaks havoc in our immune system, in our respiratory system, in our circulatory system varies between individuals. A lot of it has to do with how our immune system will initially respond to basically recognize and fight the virus, and how will it respond after that initial contact to successfully eliminate the virus and mount an immune response. And in that case, actually, you don't want either too little response, where the virus simply takes over, or too much response, where you basically have a cytokine storm and other sort of complications from fighting the virus that has been, in fact, a leading cause of death.
Okay, yeah, that's great, Manoli. And of course, each one of us is a separate case. So we keep learning as this epidemic keeps going. And this is a big difference actually with epidemics in other times, like we discussed, like the flu, because now we have the ability to gather all this information and this collective experience using new technologies and advance our fighting against the virus. And let's go back to the virus now.
Let me comment a little bit on what you just said. I mean, I just want to sort of praise the whole scientific community for how fast and how open it has been. Basically, you see across industry and academia, everybody posting papers immediately on medRxiv and bioRxiv, people sharing knowledge, sharing their insights. The genome was first sequenced in late January. Moderna had a vaccine design 10 days after that. And the fact that they have now been through phase 1 and phase 2 clinical trials in the span of months is not just unprecedented in human history, but unfathomable just a few years ago. The fact that we have gone from not knowing what is this mysterious disease that is striking citizens of Wuhan in China, versus actually understanding that this is a new coronavirus related to SARS-CoV-1 that we experienced in 2003, that its genome is a blend of different species, that the how the spike protein functions and sort of how it attaches to the genome, how to the cell of the host in the ACE2 protein, to sort of disseminating massively, rapidly information for vaccine design, for new therapeutics, for understanding what drugs work and what drugs don't work. Of course, with setbacks, bad actors exist, unfortunately, and we saw some examples of studies being retracted after falsely claiming to have results which might not be false, but at least were not supported by these studies. So there's been this incredible explosion in scientific collaboration, and I think that is why this pandemic will be very, very different from the 1918 pandemic, because we have at our disposal all of the tools of science. So yes, the first response was to massively shut down and go away from the virus, but that phase is over. We are clearly reopening throughout the world, and the next phase is really going to be using scientific understanding over the last few months to transform the way that we fight this virus and reduce casualties dramatically compared to what we would have without this incredible scientific collaboration and sharing.
I agree, Manolis. I would like to share the optimism here. And let's go to, let's focus now on this piece of genetic material that it is this virus. Tell us from your side, because you are one of the experts that can talk about this, what you can share that about the virus that makes it more, brings it closer to all of us, not from the disease aspect, but as to understand what is this this genetic material that enters the organism that can cause so many different, come up with so many different manifestations and so many, and the variety of responses.
So I am a genomicist, so I would like to start with the genome, and I'd like to start with a picture that I think has not been in the news enough, and it is this picture. So I hope that you can actually read the letters, and it is both stunning and worrisome that these 29,903 nucleotides that you see in front of you is basically what has brought the entire world economy to a halt. So this is the entire genome of SARS-CoV-2, the virus responsible for COVID-19. So you can see here 29,903 nucleotides, and I like to say of trouble, packed in a single positive strand RNA. And I think the biology underlying this virus is completely fascinating. So how can this tiny little thing that you can write in a page wreak havoc? Well, it of course encodes proteins. So on the next slide, I'm going to highlight the proteins of this virus. And here they are. You can see NSP1, NSP2, NSP3. You can see the spike protein S that everybody has been talking about. This is the protein that first attaches to the ACE2 receptor of human cells and enables the genome to enter. Its first half, S1, is basically the attachment, and the second half, S2, is allowing the entry of the virus and the release of these 29,923 nucleotides that basically enter the genome. You can see here NSP4, NSP5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. And then again, as I mentioned, spike protein. Then you have ORF3A, and then an overlapping ORF3C, which I'm going to talk shortly about. You have the envelope protein. You have the membrane protein. You have the ORF7 here, 7A and 7B, which are partly overlapping. ORF8, which is extremely rapidly evolving. The nucleocapsid protein that basically wraps up the DNA, the RNA of the virus. And then ORF10, which is actually not a protein, as I'm going to talk about later. So the first thing that you'll notice is that most proteins that we know and love are starting with an ATG and stopping with one of three stop codons, TAA, TGA, or TAG. And this is true for all of the proteins in the third part of the genome. So E starts with ATG, stops with TAA, 7A, ATG, and then ends here in this overlapping segment with TGA, and so on and so forth. ORF8, ATG, stops with TAA. There's an exception for the first part of the genome. You can see here, of course, it starts with an ATG, and it ends with a TAA, but you don't see any ATGs, any start codons or stop codons in the middle here. This is basically one giant protein. It basically makes 16 different peptides from a single translation start here, running through and then cleaving them into individual pieces after that first translation event. After that, you basically have independent transcripts packaging most of these, except for a few of them that are translated from the same mRNA. So it's a feat of engineering how you can pack so much protein coding material in so little space. If you look at the black letters, these are the only ones that are not encoding protein. So 6 letters here, 12 letters here, 30 letters here, and so on and so forth. 6 letters, 12 letters, and so on and so forth. And then, of course, the end of the genome and the beginning of the genome. So it's remarkable just how protein coding this is. By contrast, if you look at the human genome, only 1.5% of our DNA codes for protein. In this particular case, 90+% of the DNA codes for protein. Now, of course, we know a lot about this genome. So you would say, what is there to be discovered in here when things are so compact? Well, it turns out that there's a new gene, ORF3C, lurking in a separate reading frame of ORF3A. So ORF3A translation starts here with ATG, and then a little bit later from the same transcript, a new protein gets made. And this protein we didn't know about until a few weeks ago. This is a completely novel protein that we have discovered, we and 2 other studies have discovered independently in the SARS-CoV-2 genome. This protein here, ORF10, everybody has been studying it for a long time. It turns out it's not a protein at all. So we have basically discovered in my group that this is not coding for a protein. Similarly, within nucleocapsid, there's an alternate reading frame that encodes 9B and then yet other gene, another protein basically, predicted to be encoded in the same reading frame, ORF14. ORF14 is not real either. So there's still a lot that we can learn. And how do we learn all that? We learn all that through the lens of comparative genomics that I mentioned earlier. So I'll tell you a little bit about that. But before I get into that, this is basically what's causing all the trouble. Basically, all of these little proteins are encoded side by side, packaged up in a single mRNA, and then deposited inside the human cell. And these are their functions. Again, the spike protein attaches to ACE2 receptor. ORF7A cuts tethering and allows more virus to escape. ORF3C is the new one that we just discovered. It has a viral protein transmembrane domain. ORF3A cuts the cell membrane. The membrane protein actually creates the outer coat of the virus. The nucleocapsid packages and protects the RNA genome, which is otherwise very unstable, with a very long spiral, wrangling, coiling RNA. ORF14, as I mentioned, is not a protein. ORF7B, we have no idea what it does, but it's clearly a protein according to our analysis. ORF6 blocks the signals to the immune system. This is the polymerase gene. This is what creates the new copies of the virus. The helicase gene unwinds the RNA genome. This is an exonuclease, which actually corrects the genome errors. This makes RNA copies. This one is cutting loose other NSP proteins. Basically, this is the cleavage protein that cuts this giant peptide into all of these parts. So again, as a genomicist, I am, in awe of this incredibly compact genome. And the other thing that's very cool is how the viral RNA basically tricks your cells. It tricks human cells into translating a single messenger RNA into approximately 30 proteins. So this is the same information as the previous slide, NSP 1, 2, 3, all the way to 16. And then S1, S2, the 2 pre-cleavage and post-cleavage parts of the spike protein. And then the envelope, the membrane, the nucleocapsid protein, and then the rest of the ORFs. So how are they translated? I mean, most human genes translate into a single protein. Most human mature mRNAs translate into a single protein. So in this particular case, the virus, you could say, well, of course it's a virus. It could do all kinds of weird things that humans don't do, but no. This virus uses the human translation machinery to do its damage. So how can the human machinery translate something so complex? And the answer is very, very cool. So basically, first of all, as I mentioned, the first translation event leads to a single massive protein that either ends here, and that's ORF1A, and then gets cleaved into 11 peptides, and that is a marvel by itself. The second marvel is that about some fraction of the time, it basically continues translation because of a frame shifting event here between 11 and Pol. So they both have the same ATG and the same start, But then Paul, after about 10 codons, has a translation frameshift, which basically allows translation to continue all the way to here. And then these also get cleaved thanks to this cleavage protein. But then what happens to the rest? Human machinery will never translate an mRNA from the middle. So what the genome actually does is that it has a reverse transcriptase that then goes and uses an RNA template to basically create a reverse complement of the positive strand RNA virus into a negative strand RNA virus, which then, as it's translating, every now and then picks up this sequence and then jumps over all the way to here and creates a new subgenomic RNA, which is basically allowing a new mRNA to be presented to the human cell, whose translation would normally start here and basically translate into the spike protein. And then the same thing for 3A, for E, for M. These are all subgenomic RNAs that get created and result into the diversity of proteins in the viral RNA that basically make up all of these components. So our goal basically has been to now use all of this information through the lens of comparative genomics to understand the mutations that have happened. And I hope that this very short introduction gives you a handle for understanding what's on everybody's question. Yes, the SARS-CoV-2, the question that's on everybody's mind, which is, yes, the SARS-CoV-2 genome is still evolving, but which ones of these mutations matter? So in this particular case, you can see that there's been a few mutations that have been in the news a lot. So for example, a D614G mutation that basically changes a D amino acid into a G amino acid in this particular position of the S1 part of the spike protein. This is the pre-cleavage part that attaches to the ACE2 receptor in human cells. So how does this actually happen? How is this mutation rising in frequency, and why is it rising in frequency? And what about that other mutation? Well, it seems to be actually in all of the same genomes, all of the same isolates of the strain of SARS-CoV-2, and it sits in the polymerase gene. And then there's other one that sits in this very fast evolving NSP3 gene, which is as fast evolving as the spike protein. And then it also has a 5' UTR mutation, which is also evolving rapidly along with it. Sorry, let me turn off my printer.
Okay, maybe.
So, yeah.
No, please.
So basically, you can see in this single view which countries have which ones of these mutations and how far spread they have been, and in what genomic context are the new mutations arising. You can see here non-coding mutations. You can see here protein coding and protein altering mutations. You can see in blue synonymous mutations that are not having an impact on the amino acid translation, and how they spread across different continents in Asia, in the United States, in in Asia, in Africa, in Europe, in North America, and so on and so forth, here on the left. And then here, the individual countries where it has been spreading. And if you look at the D614G mutation, this has risen in frequency dramatically across multiple different populations. In Italy, as well as in the US and in other parts of the world, this has basically gone from a 10% frequency mutation to, as you can see, a 70% frequency mutation. So this is probably the sign of selection. So what we can do now is use comparative genomics to understand, is this a new mutation? Is this something that's been happening a long time? Is this changing an amino acid that's otherwise conserved, and so on and so forth. So anyway, I can keep going, but I don't know if you have any questions on the genome function or the recent mutations.
No, that was actually a very good wrap up of the very complex information that definitely make it very clear how organized this genomic material is, and is organized to survive, but also survival depends also on aggressiveness, and we are on the other side of these actions. So yeah. I would like here to get some relevant questions to the last subject you discussed. And we have from Alekos Drosos, can we make COVID virus mutate to oblivion? I will put together 2 or 3, and you can answer to all of these together, Manolis. And the other is from Konstantinos Drosatos. Do you anticipate that there will be genomics-related questions about SARS-CoV-2 and COVID-19 that will be of interest when a vaccine is available and or herd immunity is achieved? I think that we will skip the last part of the question about herd immunity for now, but talk about the vaccine. And also another one, given a vaccine from Konstantinos Antoniou, can we say anything about possible mutations? After mutations, are we back to square one or business as usual and update the vaccine like with the flu vaccine?
These are all fantastic questions and related very much to this picture. So basically, the first question is, what have we made a vaccine against? So basically, what Moderna has done is created a packaging of the S protein that basically had the ancestral form prior to the D614G mutation. So one could argue, yes, when you put in this ancestral form, maybe the organism, namely, so let me just explain a little bit how the Moderna vaccine works. So basically, it's an RNA-based vaccine. What does that mean? That means that instead of, so how do vaccines work in general? Vaccines basically work by introducing a foreign object that will then be recognized by the genome, by the host cell as non-self, and therefore mount an immune response against that non-self. The way that this is done, for example, in an attenuated virus is that, yeah, you basically take away the things that make the virus really harmful, and then the rest, well, the body can choose what it mounts the defense against, and it has many choices. And in some cases, the vaccine will work very well because it'll pick something that will be, you know, recognized across many different strains of that virus. In this particular case, the Moderna vaccine could indeed target the rest of the S1 protein without the D614G mutation, and it might work very well. But what if it doesn't? How do we update the virus? And that was the vaccine, and that was one of the questions. Well, the beauty of the Moderna vaccine is that as different mutations rise to prominence, you can basically just simply update the machine, update the vaccine, because that vaccine comes with a lot of packaging information, a lot of delivery, fine-tuning, a lot of manufacturing to make tons of it, and so on and so forth, to make millions upon millions of doses. But in the end, you can just swap out the original mRNA with a new mRNA that now has that mutation, and now make a vaccine against this mutation. This is not the same for other vaccines, but for at least this particular technology, updating it is very easy. Monoclonal antibodies is another way to go, which is basically sequence from a person the immune cells that produce the antibodies. So instead of sort of taking the serum of that person and then splitting it among, I don't know, 10 people if you're lucky, you now take the instructions for how to make more of that serum, and then you can make millions of doses. And again, if that serum was against this mutation, sure, you will have more ability to go after that, you know, in much more efficiency. So, yeah, I don't know if you know, should I go through sort of more of the comparative genomics evidence, or should I, you know, I mean, let me show you how this D614 mutation looks like across the, you know, the lens of comparative genomics. So first of all, what did we do? We basically built comparative genomics tools to analyze how is this genome evolving across related species. And you can see here, if you look at nucleotide level constraint, there are pockets of rapid change. So for example, as I mentioned earlier, NSP3 is rapidly evolving, and you can see here the pockets of rapid evolution. The S1 protein, especially the first half of the S1 protein, is super fast evolving. You can see here the evolutionary conservation drops dramatically in the first half of S1, and it also drops dramatically in ORF8, which the function of which we still don't fully understand. But if you look at protein coding evolution, we've basically written tools that specifically recognize protein coding evolution. And you can see here how this protein coding evolutionary signal is reading frame specific. You can see here that it's in frame 2, and then shifts in this frame shift event into frame 1, where the rest of these proteins are translated. And then it's basically frame specific for each of these proteins. And you can also see, for example, that ORF8, even though it is very rapidly evolving, it has a very clear protein coding signal. You can see that ORF10 here at the end has absolutely no protein coding signal, suggesting that it is in fact not a protein coding peptide. And you can also see overlapping constraint. So we can now use this information to start annotating the mutations of the virus to basically understand how it functions. First of all, we can use it to recognize protein coding genes because they evolve in very characteristic ways. If you look at the correct reading frame, then the true protein coding genes evolve in very recognizable ways. And we can use that across the phylogeny of these viruses to basically understand which mutations matter and which mutations don't. So again, just as an information piece, this is the SARS-CoV-2 genome, the Wuhan seafood market pneumonia virus. This is the SARS-CoV-1, also known as SARS-CoV classic, the coronavirus genome from the 2003 expansion. You can see here, for example, that somebody says, oh, great, they engineered a relative of SARS-1 to basically make SARS-2. Well, no, not really. They didn't. This is a very distant genome. No one would have chosen that genome if you wanted to engineer a human pathogen. You can also see that most of the relatives of that family are simply not infecting humans. They just infect bats, very happily, but no human transmission so far. But many of them may actually be the next pandemic. You don't know that. These are the rest of the human infecting viruses in that phylogeny. You can see here MERS, the Middle East respiratory syndrome, and then these four are just common cold. And they're not all phylogenetically consistent. Basically, it's not like all of the human ones are in the same place. So that basically tells you that any one of these genomes can simply evolve to be a human infector just by changing its S1 protein and changing just a few other places in the genome. So anyway, through the lens of comparative genomics, we can now go back and say which of these proteins are actually protein coding or not. And that's what we did. We basically found, a very strong signal in many of the named genes. We found that many of the hypothetical proteins are in fact real, except for a handful where we see this, very clear signal of no evolution according to protein coding signal. So for example, ORF10 here, which features prominently in many, many different genomic papers and analysis, is in fact not showing any evolutionary constraint. You can see here that ORF8, which has been doubted because of its super fast nucleotide level evolution, is in fact showing very clear protein coding constraints. And you can see here that ORF6, which again is not known, but is very fast evolving, is indeed evolving just like a protein. ORF8 is evolving just like a protein. And then ORF10 is simply not evolving like a protein at all. You basically see mostly red. You don't see much stretches of green and so on and so forth. The last thing that I really want to show, and of course, there's this new protein coding gene. This is very, very cool from a genomicist perspective. You basically have the ATG of 3A. And then as you're translating through, there's another ATG in another reading frame. So this is exactly the same nucleotide sequence, but two completely different protein translations. And you can see here the new ORF that we've called ORF3C is in fact exactly there within this much longer ORF8. And then you can see these pockets of change in the protein coding constraint. The last thing that I want to say is how the evolution within the current pandemic, the mutations that have happened recently, are in fact obeying in many ways the mutations that have happened across what we call the 60 million years of equivalent of mammalian evolution. So you can see that the fast evolving proteins that have very low amino acid conservation here on the left side versus the slow evolving proteins on the right, which have much more amino acid conservation, have many more mutations in the current pandemic than those. And a few exceptions are S1, which remarkably shows many fewer mutations in the current pandemic. Yes, D614G is an exception, but otherwise S1 is relatively slow evolving in the current pandemic. By contrast, the nucleocapsid protein is very fast evolving in the current pandemic, compared to what you would expect based on this relationship. And then lastly, here's the D614G mutation. This is actually a position of S1, even though S1 is extremely fast evolving. This is a region of S1 which is extremely slow evolving. And that particular amino acid change has never happened in the history of these subarcoviruses that span these dramatically large evolutionary distances, suggesting that this is a completely new human adaptation of this S1 protein. So the virus basically, quote unquote, invented something that it has never done before, likely because of its human hosts. So that mutation has now increased transmission dramatically, likely because it's a much better binder for the ACE2 protein in humans. It's unclear whether it also makes it more virulent, whether it also makes it more likely that you will die from the virus, but it does transmit dramatically faster from this. So anyway, that's where I won't show any more slides, but the big question is what happens next? So what's really interesting is that the ACE2 mutation, the ACE2 binding mutation in the S1 protein is in fact recent adaptation, which makes it much more transmittable. And there have been experiments that have shown that in isolation. But as I showed you earlier, this happens in the context of a polymerase mutation. And polymerase is a protein that has been evolving for, millions and millions of, mammalian equivalent evolution. And it's pretty well optimized, I would say. And that's sort of a detrimental mutation likely in the rate of replication. So one of the questions is, well, if that seemingly advantageous S1 mutation is only found in the context of a seemingly disadvantageous polymerase mutation, what will happen if the genome actually splits the two and is able to maybe replicate equally fast while at the same time transmitting much, much faster? So I would say it's a very young virus. All bets are off. It could mutate to become much more virulent. It could mutate to become much more deadly. Although if you look historically, viruses in pandemics only mutate to become more transmitted, not more deadly, because more deadly is not helpful to the virus. If we die too fast, we don't transmit it as quickly. And the way that you should think about this is that there are two strains competing. One strain just kills you off right away. The other strain just simply makes you mount an immune attack and it doesn't kill you. The strain that doesn't kill you will spread faster just because people will not be as incapacitated. And it will also act as a virus, because as a vaccine, because it will allow people to foresee the coming of a very, very close relative, which is actually much more potent and much more virulent. So in pandemics in general, historically, and based on these first principles that I'm describing, you generally see the virulence attenuated, but the transmission increase to the point that it might become just another common cold where you have everybody getting it, but nobody dying from it, or at least very few people dying from it. And I think this is probably where this virus is also going to be going, even though, as we can see already, it hasn't yet reached its full evolutionary capacity in a relatively young and virgin human population, which has never been exposed to something so closely related before. I hope that helps answers these questions.
Yes, and now this was very informative, actually, of how the virus finds its way to do what every life does, actually, to maintain itself. And so let's go to the human side. We have two questions here that are relevant, that from Andreas. What about the risk of antibody-dependent enhancement? So antibodies created through vaccination may possibly make it even easier for the virus to enter the cells. Can you put it in the context of what you shared with us about how the virus works and the proteins that it makes? And also, if there are antibody tests available, yes, there are, but what is the best way to find? I suppose also the question is, are they good enough to tell us if we already had the virus? And it is to be seen if this immunity lasts for how long. This is actually subject.
These are fascinating questions. So basically, the first point that I want to make is that detecting if you have the virus basically is an RNA test. It's a PCR reaction, it's polymerase chain amplification, which basically simply test is if the virus is actively replicating inside your cells. And this basically only works in the first 10 days or 14 days of infection, basically, or, you know, I would say from day 3 or so to day 14 or so, when there's enough virus and it's actively replicating, but before you get really, really sick and your body starts either absorbing it or killing it or, you know, fighting with it. So the RNA or the nucleic acid test is very reliable because you can detect infinitesimally small amounts with extreme precision because of this amplification that you're doing. Antibody tests are much more difficult for everything. It's not that, oh, this year we have really bad antibody tests. It's just a more difficult test to do. And that's why we don't yet have very, very reliable antibody tests, and the false positive, but also the false negative rate is actually pretty high. So unfortunately, this is what we're stuck with. And knowing whether you've had the virus or not is probably going to be a combination of questionnaire of have you had related symptoms and also just testing for antibodies. The other point is that many people are saying, oh, great, the antibodies are going away after 30 days or something like that. Well, yes, the antibody life itself is fairly short, but the cells that make these antibodies are still in your body. So just because you're not testing positive for antibodies now, simply probably means that you've been infected, you know, more than a month or 2 ago, and that's why your body is not actively presenting these in your blood. But chances are that you might still be immune because your immune repertoire has now saved the cells that can make more of that antibody. As for antibody-dependent enhancement through vaccination, this is sort of related to this whole concept that if your cell now has a receptor that is very well tuned to the spike protein through an antibody, maybe that cell would basically go actively seek out the virus and bring it to the rest of your immune system. And yes, I guess this has been an issue with some viruses, but I think this is such a minor concern because the alternative is the ACE2 receptor already has an amazingly awesome affinity to Yanni, please την πόρτα τώρα παρακαλώ. Αγάπη μου.
Είδα 2 αυγά μέσα σε ένα νύχι.
Τι ωραία. Μέσα στην αυλή έχουμε 2 αυγά από πουλάκια. Μπράβο, αγάπη μου. Οκ, λοιπόν, έχω 3 παιδιά όπως βλέπετε. Και τους αρέσουν τα πουλάκια και η φύση, οπότε ελπίζω να γίνουν και αυτοί οι επιστήμονες. Sorry, I switched to Greek. I was basically saying that the alternative, basically, there's two options. One is the virus basically finds your ACE2 receptor and gets in through there, and that is already an almost perfect affinity. And then the alternative is, well, somehow your immune cells that will most likely destroy the virus also bind to it well. So I would say that the cost benefit is much tilted on the way of, no, the therapeutic is much more likely to be effective rather than to lead to a stronger infection.
Yeah, I agree. And actually, although we are not experts, I think that on the antibody generation and maintenance, we both agree that it will take time and we need much more information. And but it was very clear from what Manolis shared on the genome of the virus how this process works. And I will take now 2 questions that are related, are not so specifically scientific, but they are at the core of this epidemic. And one is like, we have a question that it is, excuse me, one moment. The one is about the increasing cases that we see in the South and West. What would be the advice for them? And also if CDC and WHO have been as effective as possible. And I would like for the second question, if you can reply from your side as a genomicist.
So the first question is about the South and the West and the increasing cases and what advice I have for them. So basically, I would say follow the advice of scientists. They know what they're talking about. Basically, wear masks, stay away from large gatherings, stay outdoors. If you want to meet your friends, do so at some distance in a garden. enjoy the summer, don't stay indoors, don't stay in crowded places, don't take public transportation, don't share the bathroom with anyone else. I mean, sorry to be so blunt, but as the virus can actually appear in feces, when you flush the toilet from a person that is infected, it aerosolizes viral particles. And that basically means that the next person who enters the bathroom, even if it doesn't smell, has basically the full repertoire of viral particles that was in the feces of the person who entered before. So don't use public restrooms. If you go to a restaurant and you're wearing a mask on the way to the bathroom, don't take off the mask when you get in the bathroom. That's the worst place. You should always wear the mask. So advice, masks, surface contamination compared to many other viruses doesn't appear to be a major transmitter. So if you have only a limited budget, spend your budget in isolating workers from respiratory transmission rather than spend your budget on sort of wiping surfaces clean all the time, because that appears to be, at least according to current knowledge, less of a form of transmission. And related to the advice that I'm giving now, has the CDC and the World Health, the WHO, the World Health Organization, been as effective as possible? The answer is obviously not. Initially, the reports from the World Health Organization were that there is no human-to-human transmission. And many people went about their business thinking that there is no human transmission. The second mistake that they made very early on was don't bother with masks. Both were mistakes. Are these malicious, or are these simply because science is an evolving field and we're learning more as we go? My guess is that the second one is probably partly malicious, and I wouldn't call it malicious per se, in the sense that there were dramatic mask shortages. So there was a conscious decision from health organizations to basically tell people don't buy masks in order to save them for the health workers. I think it was a mistake, and I think it was an error, and I think it was a conscious error. And the alternative would have been to just recall all the masks and take them back from all the stores and give them to the first responders, because that was the right thing to do, rather than sort of trick people into thinking, oh, masks are not good for me, and then reverse the decision, leading to less trust in the scientific authorities and the World Health Organization and so on and so forth. So that was clearly a mistake. As for whether it's human-to-human transmission or not, I don't know. There's a lot of conspiracy theories about that. I would say that they did what they knew at the time, and maybe they were influenced by China, as have been a lot of reports. Maybe they were not. It's unclear. The main thing that I want people to take away from my very convoluted answer here is that scientific knowledge evolves. Scientists are not always right, but at least we can trust them to do, with the best of the knowledge that they have right now. So I don't believe in conspiracy theories. Instead, believe in scientists and look at multiple scientific forums to get a formed opinion. If the World Health Organization says one thing and your local authorities say another thing, look for a third opinion. Look for, sort of more guidance from scientists. Look at blogs from well-reputed sources and try to get more information in those places. But basically, the current scientific consensus is absolutely wear masks, absolutely, wash your hands, always apply hygiene, avoid bathrooms, avoid closed areas, and stay open as much as possible. I hope this helps. I'm not mincing my words, and I'm not hiding the fact that there were a lot of mistakes that were done in the current. And if you look at the CDC, the original tests that were sort of sent around in the US were flawed. So there was also a guideline that you were not allowed to sort of develop your own test and start giving it to people, because they needed to sort of be the providers of genome-based tests. Yeah, these were mistakes. There were errors made, and I think that has led to a lot of the crises that we're looking at now in the United States. Basically, a slow response, less communal wearing of masks, and less isolating due to a combination of many causes. And factors, and also mishaps by the CDC that was not, was basically unlucky with its tests and not as effective with its advice. I hope this helps. Yes, there were mistakes.
Yes. Yes, thank you, Manolis. So we need to wrap up soon. So I would like to put together two questions. One is about the transmission through the cold food chain, and the other is from the Good News Greece, the collective knowledge that you so articulately shared with us will certainly help build the vaccine. Can it help also in the development of a drug or a cure? But if you can be very short on that and just go to the very important question that how about Greece and this epidemic? We know that they gave an amazing example of how disciplined all Greeks were in staying at home and actually protect the elderly and the more susceptible people, but also we saw that it was a fantastic effort scientifically at a level that we could not dream of, because it was so massive and so enthusiastic and really very contributing for the world. So what we learned from that, and what could be the next step? How can we all contribute help to, if as needed, to keep it up and for all that is coming?
So, I mean, these are extremely difficult topics, and I don't think that I have all the answers. I mean, the future will tell. It's very difficult to make predictions, especially about the future. But my fear is that Greece is a very virgin population right now. We have not been exposed to the virus. We're not Sweden. We're not Italy. We're not New York. There's a tiny little percentage of the population that has actually been exposed to SARS-CoV-2, and therefore the herd immunity level is nil. There's we're basically still at square one when it comes to the virus spreading. So that basically means that if there's a wave of carriers arriving in Greece starting July 1st, we will be in bad shape. This will spread the same way that it spread in all of the other countries. Modulo, of course, the immune repertoire of Greece, shaped by all of the infections that we have survived through the plague, through the, history of European infections, through the, 1918 influenza, etc. All of these have shaped the immune system of different countries in different ways. And we have passed on this inheritance of the subset of people who survive these massive infections for each country. So every country has its own immune system. So maybe there's an aspect of immunity in Greece that is innate and running in the country. I shouldn't be using innate in a scientific context, where it's an adaptive immunity that is sort of built into the country. That could basically reduce perhaps the impact that this new influx of mutations may have in Greece, and may have also contributed to the reduced impact of the mutations in the first round back in March. But my thinking is that we're going to be facing our first wave as the tourists start arriving. And that basically means that we have to not ignore masks, we have to not ignore guidelines, we have to keep our distances, stay in open places, wear masks as much as possible, do not and the more you speak, the more viral particles you shed. So Greeks like to speak a lot and they like to speak loudly, and it's a cultural thing. And we should not be facing each other when we speak. We should be keeping a distance. We should be wearing a mask, covering our mouth. We have to sort of be cognizant of the fact that I'm protecting others and I'm protecting myself as well, but mostly protecting others through my actions. Even if I'm young and healthy, who knows if my friend has parents with, who are elderly, or siblings with pre-existing conditions, or loved ones who are at greater risk, etc. So we have to stop the spread as much as possible. So I would say the advice to Greeks in this new phase of reopening our frontiers is going to be pretend that we're on the first wave in March and keep your distances as much as possible, because the alternative is the virus spreads and we have to lock down again, and nobody wants that, and we don't want to be there. So keep your distances as if this was March, and stay far apart from each other. That's sort of point number one. Point number two, I think all of the authority guidelines of sort of quarantining people, testing, I think there's a major question of enforcement of these quarantines and sort of how do we test that. Cell phone based tracking could be one way. checking in, random tests, cameras, you name it. Like, we have to basically be very strict. Basically, if we want to protect the entire population, we have to give up some luxury of the tourists coming in with, protections appropriately placed. The other question is, how can this knowledge be used in the development of a drug or a cure? And I think for this, we've talked a lot about vaccines, and there's many different strategies of vaccination. On the cure and the drug side, the repertoire of drugs that we have against fungi is great. There's a bunch of antifungal. Against bacteria, great. There's a bunch of antibiotics. Against viruses, as you saw, there's only 29,000 nucleotides. There's only like 16 proteins that you could potentially target. Which one do you target? Which one mutates the slowest? How do you build a drug against this? And there aren't right now any large-scale antivirals. What would they look like? They would probably target the replication machinery, which is shared by a lot of viruses. The problem is that it's very fast evolving. It's very difficult to sort of make a wide-ranging antiviral. There have been a few cases of, for example, steroids have been shown to decrease this sort of inflammatory response that sort of is associated with a lot of deaths. So I think, steroids is one answer. Hydroxychloroquine is, of course, not an answer. It's actually been shown to be more detrimental than helpful. So that's unfortunately out of the picture. Or I should say, fortunately, out of the picture. There are very simple steps like placing the patient, facing forward. That makes it easier to breathe, much less use of a respirator. So there's basically standards of care that we now know. And I think that will make a dramatic impact. And then, of course, there's massive amounts of effort in repurposing existing drugs for either other viruses like Ebola and, you name it, where there's, some hope that we will see more effectiveness there. So we're not in the same situation that we were in March. If you do get infected, there are much better guidelines for how to treat people and how to protect doctors from spreading it within the hospitals. And then the last question, and I don't remember it anymore.
It is related to the Greek science, the Greek scientists, and the Greek scientists from abroad.
So I would say that what the pandemic has taught us is that the world is actually a very small place. It makes very little difference whether you're in the US or in Greece or in France or in South Korea. We're all in this together. That basically the guidelines that we've learned from one country apply to another country, and sort of spreading and sharing that knowledge is extremely important. But it also taught us that the world is a small place when it comes to communication. At the thesis defense of my student, there were 20 family members from across the world that would not have been able to actually physically travel to be at her thesis defense, and yet we're there. We have Zoom birthday parties where basically people come from, across the world, from many different time zones and many different continents. And in my research group right now, I have a scientist who's in South Korea who calls into our group meetings, a scientist who's in California, and a scientist who's in France. We're basically around the world, all in the same one-on-one or one-on-many group meeting and conversation. So that means that collaboration is at an all-time high. Instead of flying for 3 hours to go have a 1-hour meeting in New York, or flying for 10 hours to go have a 1-hour talk in Paris, I can just show up, have the meeting, have a bunch of one-on-one conversations, and then speak in London 3 hours later, and that's perfectly fine, and that's perfectly feasible. In the same way that I speak with my own lab members. Of course, human contact is extremely important, and I encourage all of you to sort of pick your bubble, pick your set of very small number of people that you want to have close interactions with, social interactions outside your immediate family circle. Make sure that they're also only in your bubble and not in a dozen other bubbles. And then have those social interactions. But in terms of scientific collaboration, the frontiers have been, abolished. The borders have been abolished. And I think we have an opportunity. I think Greece, in the context of this Hellenic Innovation Network, and in the context of Enterprise Forum Greece, and in the context of all of these amazing initiatives that Stratos and Marina and Vasilis have put together, we have an opportunity to basically turn Greece into a hub of collaboration. We have the opportunity specifically for genomics and for computational biology. I mean, we heard a lot about the challenges of biotechnology, where you require a lot of infrastructure. But for computational biology and for genomics, the stuff that I've shown is accessible to a huge number of Greek scientists who have computer training, mathematical training, informatics training, and so on and so forth. And I see this as a next frontier for Greece. Yes, we're in a rush to reopen our borders to basically keep the tourism economy alive, which is extremely vital to Greece, of course. But at the same time, I see this pandemic as an opportunity to sort of push for more informatics training, for more remote collaboration. I'm now setting up several collaborations with Greek teams that allows us to communicate over Zoom. And I'm even looking at hiring people in Greece through a company that one of my friends set up called Globalization Partners, which basically allows me to hire a person as if I was hiring them here, to have them on my payroll within 3 days, because Globalization Partners has an office in Boston and it has an office in Greece. And I can basically pay the office in Boston, who then pays their subsidiary in Greece, who then does all of the paperwork for actually hiring the Greek scientists in Greece. And within 3 days, I can have someone on my payroll instead of fighting with H-1 visas, which have been unfortunately, suspended until the end of the year, without having to relocate, without having to now take whatever income they make and spending it outside Greece, which of course decreases the impact on the Greek economy. My goal would be to sort of look at such solutions where Greece could basically set up an employment hub with an office in Greece, which can now set up parent offices in many different countries across the world to enable Greek scientists who want to work in France or in London or in Germany or in Asia or in the US, of course, to basically have mutual agreements of partnerships where I can basically hire Greek scientists as part of my team, effectively treating them as consultants or subcontractors through a Globalization Partners like setup, but now centered in Greece rather than centered in the US, and from Greece going out, to basically enable Greek graduates to have zero problem with employment anywhere in the world without actually having to travel. Because frankly, if you're going to choose a place to live, I can't think of a better place than Greece if you can work anywhere in the world and live anywhere else in the world. So I would love to talk with Marina and with Stratos and with Vasilis about sort of setting something up that allows this diaspora of Greek minds without the bodies, where you can sort of keep physically the people living in Greece while their brain can be interacting with scientists across the world. And this abolishment of borders and frontiers that the virtualization of collaboration has allowed us can be translated hopefully into a very large economic impact and very concrete steps for both training a generation of data scientists in Greece, and then having them rise to the occasion of addressing data science problems that are pervasive in the world right now, across transportation, across social networks, across politics, across all fields of astronomy and space science, and of course biology, genomics, comparative genomics, epigenomics, etc. The entire sort of realm of biology, which is now amenable to dramatic need for data scientists to integrate all this data and gain new insights. So I'm very hopeful about the role that Greece can play. We have an outstanding generation of very strong minds who have been leaving Greece to find opportunities abroad. Maybe we could sort of see a next phase where they don't have to actually physically leave Greece. They can still live in Greece and sort of collaborate with the world. And I hope that Greece can play a very big and fast role in creating such an ecosystem.
Great. And with this, we are a little bit over time, but it was a very informative and very insightful discussion with an expert. And thank you very much, Manoli. And I would like actually Vasilis or Marina to take over. wrap up.
I want to also add my thanks to Marina, Vasily, and Strato. I mean, I have been blown away by the quality of these webinars and the conversations. And I want to thank each of you because you are beacons of hope. You are beacons of energy and drive that can do so much. And I'm truly humbled to be among all of you guys here because you're doing so much for the Greek community. And we are all so proud of what you've accomplished. And I think the whole series of seminars has been truly an amazing piece of work. So thank you all.
Thank you. Thank you for that incredible talk. We learned so much. Really amazing. And you actually touched on something very relevant as we resume our series in the fall. We're going to be talking about journey to exit and innovation in agriculture as we move beyond COVID. But you touched upon something that we would like to discuss that is in Vasily's heart, which is digital nomads. How can we create a community in Greece of people who are working in Greece and delivering their product to places all over the world? So I don't know, Vasily, if you want to talk about
Love that. Basically, again, I was talking about Greeks who don't have to leave anymore, but no, this can be the whole world moving to Greece and then not having to move anymore. I love that.
Yeah. Yes.
Yes, I think what you, Manolis, pointed out is very important, and it's a great opportunity. Maybe the new globalization is not about blue-collar workers as it used to be and going to low-cost countries, but it will be more about people, scientists working around the world and finding better places and to live at better quality lives. And I think, yeah, in Greece we have a lot of bureaucracy and a lot about stuff, but this is not about working in Greece, it's about working from Greece, which makes a big difference. Thank you for the comments you put together. As Marina said, this is closing the first part of our discussions, and we are getting ready for a more organized series of webcasts starting from the fall, and we will be back. Strato.
Ένα μεγάλο ευχαριστώ. Thank you very much, Katia and Manolis. We're really privileged to have you among us and among our friends and among our scientific community. It's because of you that this scientific community is elevated and is the best Greek scientific community in the world. Thank you once again.
Thank you so much. Thank you and the many others who contribute to making this community awesome.
Thank you. Signing off.